Bismillahirrahmanirrahim, and welcome back to this next video. And this is the part 2 on the Alkaptonuria. In my previous video, I've told you that Alkaptonuria is an example of an autosomal recessive disorder, and during this particular condition, there is a buildup of a particular metabolite which is known as the homogentisate acid when you go for the metabolism of the tyrosine and phenylalanine amino acid. And this buildup is because of the deficiency of an enzyme or the non-functional copy of an enzyme which is known as the homogentisate dioxygenase. In this particular video, I want to focus on the genetics of the Alkaptonuria. Now, as I've told you in my previous video, that Alkaptonuria is an autosomal recessive disorder. So, what do you mean by an autosomal recessive disorder? So, if you look at over here, there are two terms. One is known as the autosomal, the other one that is known as the recessive. So, let us understand this. The autosomal mean that the gene is present on the autosomal chromosome. So we are talking about the gene, we are talking about the enzyme homogentisate dioxygenase. So that particular gene is present on an autosomal chromosome. In this particular case, it is present on the chromosome number 3. Secondly, if you talk about this recessive term, it means that both copies of the genes, the one that you are inheriting from your mother, the one that you are inheriting from your father, so both copies of the genes, they are mutated, and both of these mutations, mutated copies of the genes, that actually leads to an affected person. If you talk about the alkaptonuria, as this is an autosomal recessive disorder, so a person who carries one copy of the mutated gene and one normal copy of that particular gene, they are said to be the carrier for the condition. For example, if you are getting a normal copy from your father, you are getting a mutated copy from your mother, you will be a carrier and you are not going to show the symptom of the alkaptonuria. Or otherwise, if you are getting a normal copy from your father and a mutated copy from your mother, again you are going to be a carrier and you are not going to show the symptom of the alkaptonuria. If you talk about the gene, that is known as the HGD gene, and this is going to code for the enzyme, the homogentisate 1,2 dioxygenase. So the gene involved in the alkaptonuria is the HGD gene. If you look at this particular gene, it have 54,363 base pairs. It contains 14 exons, and it makes up a protein that is 445 amino acid long. So this is the general information about the gene that is involved in the alkaptonuria. So what happens during the alkaptonuria? Why this particular enzyme is not working? Why there is a deficiency of this particular enzyme? And the culprit, they are the mutations. So a lot of mutations, they are there in the HDG genes, and these particular mutation is going to lead to a non-functional or a low activity showing HGD protein. So let us talk about some of the reported mutation in this particular gene. So if there is a mutation in the HTG gene, the enzyme that it produces, that could be either non-functional, you will see that in the examples, or that particular HDG that produce the enzyme that have a significantly reduced activity. And over 100 different mutations they have so far been identified in the HDG gene that is responsible for the alkaptonuria. So let us look at some of the examples. The first type of mutation that you see in the HGD gene, that is known as the missense mutation. And when we use the term missense mutation, that would mean that one amino acid is swapped for another. What I mean by that is that in a normal HGD protein, you are going to see one amino acid, and when there is mutation, instead of that particular amino acid, you are going to see another amino acid. This is what the missense mutation mean. Let us look at some of the examples that is going to give you a very clear idea about the missense mutations. This is one of the example. So the first thing you need to understand is how to read this. This C is actually referring to the coding area. So you're talking about the messenger RNA, for example. This 673 is going to show you the nucleotides where there are mutations, where mutation is going to take place. So if you read this particular one, you can say that in the coding area of this particular gene at position number 673, normally there is a cytosine nucleotide, and now that has been replaced by a thymine nucleotide. So this is what this C.673C to T means. When you talk about this particular thing, this P is actually referring to the protein. This arginine is actually the amino acid that is present in the normal HGD protein. This is 225 is going to show you the position where, because of this mutation, you are now going to get a different amino acid, and the different amino acid that you are going to see is the cysteine in this particular case. So you can say that because of the change of the cytosine into thymine at position number 673, that is going to change the arginine at position number 225 into the cysteine. So as you have swapped one amino acid for another one, this is known as the missense mutation. Let me give you another example. Now that would be clear to you that at position number 433, normally there is a cytosine that has been replaced by a thymine, and in the protein, because of this mutation, the arginine at position number 145, that has been changed into the tryptophan. Now when you talk about this missense mutation, you are going to see changes in the amino acid, but you are going to get the whole protein in most of the cases, and these particular protein are going to show some reduced activity. Their activity is not totally lost. That is going to show you decreased activity. So you will be saying that there is deficiency of this particular enzyme, and because of this deficiency, there would be buildup of the homogentisate acid in the patient of the alkaptonuria. So this is one kind of the mutation that you see in the Alkaptonuria. Another important example of the mutation that you see in the Alkaptonuria or in the HGD gene, that is the nonsense mutation. Now, as the name indicates, that this mutation is not going to make any sense. This is a nonsense kind of the mutation. And what happens is that in the nonsense mutation, a stop codon is introduced prematurely. So you are not going to get a complete protein. You are going to get half protein, a truncated kind of the protein, because a stop codon that has been introduced prematurely. Let me give you some example. You're already getting the idea how to read this in the coding region of the messenger RNA. For example, at position number 481, there is a cytosine normally that has been replaced by by a thymine, and in the protein, normally at position number 161, there is an arginine. So what does this steric mean? The steric actually refers to a stop codon. So how you are going to read this? Because of the mutation at 481, because of the change of the cytosine into the thymine, normally at position number 161, there is an arginine that has been now switched to a stop codon. So you are not going to get any protein sequences beyond the arginine 161. So the protein that would be very short, because I've told you that the total length of the protein is 445 amino acid. Now you are going to get a protein of 160 amino acid, because at position number 161 you have introduced a stop codon. So that is going to give you a premature stop codon, a truncated kind of the protein, which is not going to give you any kind of the activity. Another example that would be this one at position number 187, the cytosine that has been changed into the thymine, and in the protein, the arginine number 63 that has been changed into a stop codon. So you are going to get a premature stop codon at the amino acid number 63. Again, you are going to get a truncated kind of the protein, and that truncated kind of the protein is not going to give you any kind of the activity. The third and the important type of the mutation that you see in the Alkaptonuria, that is the frame shift mutation. This is another kind of the mutation that you see in the Alkaptonuria. And we will discuss the frame shift mutation and the results that it gives you with regard to the patient of the Alkaptonuria in the next video.